research focus of my lab is cancer epidemiology and cancer biology. I have been trying to understand the etiology of most of the common cancers in Kashmir, including esophageal squamous cell carcinoma. We conducted a number of studies on esophageal cancer, and we were able to identify the most common risk factors in Kashmir for this malignancy. In addition to that, I have been trying to understand the biology of cancer, including angiogenesis, migration, and metabolism. Currently, my research interests in the lab are we are trying to understand the epigenetics of neovascularization in esophageal squamous cell carcinoma, and we are trying to understand the role of NF-kappa beta in understanding the regulation of glucose 6-phosphate dehydrogenase in pentose phosphate pathway, which is unreported and unexplored yet. In addition to that, we are trying to understand the role of heat shock proteins in migration of the cancer cells. My lab is open to all the students for any position for training. Thank you.
The major focus of our research laboratory is to understand the various steps that occur during the process of tumorigenesis. We particularly study the role of various genes, especially kinases and phosphatases that have a role in the regulation of cell cycle, as well as various signal transduction pathways that have a very critical role during the process of cancer progression and development. As of now, our major focus is to also understand the mechanisms that lead to the development of anti-cancer drug resistance, which is in fact the major concern and the challenge that cancer biologists and oncologists face as of today.
Hello and welcome. My name is Dr. Ashraf Dar. I am the principal investigator in the lab of ubiquitin biology and genomic instability at University of Kashmir. Our lab is investigating the role of ubiquitin proteasome system in the regulation of different physiological pathways in cultured human cells. Ubiquitin is essentially a small 8 kilodalton protein that is tagged to the other substrate proteins by the E3 ligase enzyme. The ubiquitination of these substrate proteins acts as a trigger for the substrate proteins to get degraded in a specialized structure in the cell that we call as the 26S proteasome. There are other classes of the proteins that are involved in untagging of the ubiquitin from the substrate proteins that we call as the deubiquitinases or DUBs. So the E3 ligases and deubiquitinases, they are thus involved in the homeostasis of the different proteins in the cell. So over the last few years, our lab has discovered many cellular pathways that are regulated by the ubiquitin proteasome system. These pathways are involved in the regulation of cell cycle, DNA damage response, homologous recombination, and autophagy. So these studies are helping us to propose therapies in cancer and other killer diseases. Thank you.